guitar player Johnny Winter and his brother Hector. They both had this long blonde hair, especially if you're in a club like that had like strobe lights. Their hair would kind of like glow white. You're like, whoa, dude, that was cool. All right, let's move on from Johnny Winter. Let's now talk about another complication of Mendel's genetics. This is, well, I have to write it down. I wrote it for you right here, pleiotropy. Okay, it's where a single gene affects more than one character. Again, that's not a Mendelian trait. Mendelian traits is where one gene influences one character, where there's two alleles with complete dominance. Now we have a single gene affecting more than one character. And the classic example is sickle cell anemia. Now sickle cell anemia also shows incomplete dominance as well. Because if you think they use HB, I think N for normal, I forget the code here. So if you have that genotype, you're completely normal. You don't have any sickle cell at all. If you're heterozygous, okay, you have what's called sickle cell trait. You produce some normal. This is an allele that disrupts the gene for the beta globin chain of hemoglobin. So here, completely normal beta globin chains in the hemoglobin. This individual, here you got one correct allele. So most of the beta globin you produce in hemoglobin is normal, but there's some that's sickle will cause this disturbed shape here. What happens now? Blood cells have this sort of donut shape. They're not really a donut. They're what we say transverse. They have what's called a biconvex shape. They just tend to look like a kind of the middle's open there. This is one of the few, if the only cell in humans or in vertebrates that has no nucleus. They get rid of it after they're produced in bone marrow. It's essentially a bag of hemoglobin that has a shelf life about 120 days, produced in bone marrow, carries oxygen from the lungs to the tissues and CO2 back. Okay, and then when they're trash, you kind of ship them to the spleen where they're kind of dismantled, the heme's recovered, all that good stuff there. For the sickle cell trait, there's the beta globin, I believe, I could be wrong here, but I believe the beta globin protein is 146 amino acids long, and a position number, is it 6 or 8? Let me look at my next note here, I forget if it's position 6 or 8. What is that, position 6? Okay. At position 6, there's one SNP, one amino acid change. A glutamic acid is replaced by a valine. Now, glutamic acid, this amino acid, you can look up the structure. It has a carboxyl part of the R group, so it's a charged amino acid, or it's an acidic amino acid with a charge, makes it polar. Okay, so it's polar. Valine is a non-polar amino acid. Okay, so you got one amino acid difference at 146, just one. And if you're at normal oxygen levels, like at sea level, you're not exerting yourself, well, you're cool. Your red blood cells will be normal, you'll be okay. But if you get stressed for oxygen, you're at high altitude, you're exerting yourself, at low oxygen conditions, this non-polar replacement tends to stick to a hydrophobic patch on part of the protein, and it warps out of shape to produce this sickle. And that clogs capillaries. And you can't get blood to vital tissues, and you suffer anemia. And it causes massive tissue damage. So it's an example of pleiotropy, where one allele causes impaired mental function, heart failure, weakness, fatigue, pneumonia, heart failure, kidney failure, abdominal pain. It's what we say in my day, that's a bummer. Okay? All right? So one allele causing only multiple conditions. Okay? Now, if you're homozygous, I didn't continue this. If you're homozygous recessive for this bad allele, you have sickle cell disease. Sickle cell disease versus sickle cell trait if you're heterozygous. Sickle cell disease, if you're born in the wrong country, you're probably going to die as a teenager. It's pretty debilitating. You're super weak. I mean, these are some serious issues here. You're not going to make the top of your class. You know what I mean? All right, and you're gonna die young. Especially this gene is most prevalent in Africa and in places like around the Mediterranean where there was malaria present. It turns out that if you're sickle cell trait, you're more resistant to malaria because the malarial parasite cannot survive in the sickled cells. So that's why this very dangerous allele sort of survived through generations because it provides some adaptive benefit to those heterozygous individuals where malaria exists. Malaria is the world's #1 infectious killer. When people talk about infectious diseases, #1 killer in the world, people say, you ask students or anybody, what do you think is the #1 infectious killer in the world? People, well, it's got to be HIV or Ebola. Bullshit, it's malaria. Malaria kills about 1 million kids a year. And for decades, the U.S. government and the military have tried to produce a vaccine. It doesn't work because this parasite just evolves so fast. So, so the best strategy is to fight the mosquitoes, and mostly by insecticide treatment like DDT. Yeah, there's somebody. Yeah.
I'm sorry. I've had malaria a couple of times.
Yeah. Well, once you get it, you got it forever.
Oh, really?
Yeah. Once you get the parasite, you got it. What usually happens is that once you get your first infection, you have that cycle of fever and chills, and then usually your body responds and it kind of goes away for long periods of time. But sometimes it kind of reemerges. I know that because when I was a grad student at Berkeley, when I was doing some work in chemistry, the chemistry prof there had been a soldier in the Philippines in World War II, and he got malaria. He told me the same thing. He said at first he got real sick, and it kind of just petered out. But every once in a while he would have an episode. The dangerous part is when the malaria can cross the blood-brain barrier and get to your brain, and you're really screwed. But otherwise, it is a condition you can live with, a chronic condition. So it's a little more serious than having herpes, but it's sort of like that. Once you got it, you got it. And once you get herpes, it's a non-curable condition so far. Yeah.
So what does it do to your brain?
Well, you get a parasite in your brain, it's not a good thing. I'm not sure, but I'm sure it causes massive tissue damage. And since the nerve cells never regrow, so, you know. It's a complex life cycle. When we get to 1.1b, it lives in 2 hosts. It lives in, at least the human malaria, it can also live in birds, but warm-blooded hosts, but it lives in mosquitoes, the salivary glands of a mosquito called the Anopheline mosquito. The reason malaria is not a big issue in the temperate world is because that mosquito is a tropical mosquito. But as the world is warming, it's coming further north. Okay, so malaria is coming our way. And, but of course it's only a Chinese hoax, so we don't have to worry about it. And then the other hosts are warm-blooded vertebrates like humans and birds. Okay. All right. So that's an example of pleiotropy. One allele causing multiple phenotypic effects. Okay, let's look at our sort of last complication as we wrap up chapter 12. And that is, let's look at what's called polygenic traits, or also known as quantitative traits. Polygenic or quantitative traits. And I have to say many human traits. We think about things like skin color, body height, body weight, hair color, eye color. These are polygenic traits. And what do you think polygenic means? What does poly mean? Yeah, so it's, in this case, it's where you have multiple genes controlling one character. It's kind of the opposite of pleiotropy, where pleiotropy is one gene affecting many characters. This is where you have one gene. I'm sorry, said it backwards. Multiple genes, multiple alleles, influencing one character. So it's the opposite of pleiotropy, where it was one character influencing one gene influencing many characters. This is multiple gene influencing one character, like hair color. Or here's the example I'll show you right now. Eye color. Your eyes can go from very dark. We're talking about the amount of melanin in the iris here. It can be from very dark all the way to very light. And the clue that a trait is polygenic or quantitative is when you have a range of phenotypes. Remember a Mendel trait. It was either purple or white, either yellow or green, either wrinkled or round. It's either or. This is a range of conditions, like a range of eye color, skin color, hair color. Okay? We see that beautiful range of skin colors from very dark to very light, and somewhere in the middle. Most of humanity is sort of here in this sort of intermediate place right here. And so if you've got many genes influencing this one character like skin color, we call each gene locus, where all these different genes are located on different chromosomes, they're known as QTLs, quantitative trait loci. And a lot of complex diseases like heart disease, cardiac disease, cancers, those have many gene influences. They're all are quantitative diseases. I mean, cancers run in families, so do heart disease. So we know there's a genetic component, but there's also for those diseases an environmental component. What is your diet? Do you smoke? Do you too much sunburn? So polygenic traits almost always have an environmental influence. I mean, think about skin color. Can you influence your skin color environmentally? Sure you can. You stay out in the sun and tan more, or stay in the sun and stay out of the sun and let your skin go as light as possible. That's natural environmental influence. Other folks do it cosmetically, like the late Michael Jackson. He kind of went a little extreme, in my opinion. But there are many different genes at many different positions on different chromosomes that contribute. And in diseases like cancer, what cancer scientists do, they try to look at the genes that contribute to cancer and try to find all